Okay. Good morning, good afternoon. Today I will be talking about distributed quantitative proteomics tools. My name is Perez-Riverol. I am coordinator of PRI team, and these are my contacts in Twitter and GitHub. Um, the outline of this talk is basically first a very short introduction about quantitative proteomics tools, and a summary of the existing tools. And I guess you have seen during the course other presentations about quantitative proteomics, general background and theory about quantitative proteomics. Today I will be giving a really good talk about distributed analysis of data, quantitative proteomics data using OpenMS, and then a short introduction at the very end about cloud-based data analysis using a Nextflow workflow, specifically Proteomics LFQ. As you have seen during the course, basically mass spectrometry proteomics experiments run from basically the biological samples, and then you try to extract or separate the proteins, and then digest those proteins into peptides, and those peptides are going into a chromatogram and then a mass spectrometry when you finally capture the mass spectra. I hope you have seen the theory of all of this. This is really, really a simple workflow about what's in a nutshell is happening. But of course, there is a more complexity here, that if you have, for example, label experiments, you will have some labeling process here in the cells at the very beginning. If you, for example, if you have different separation techniques, and you can use chromatographic technique, but you can also use other other techniques, for example, 2D gels and things like that. And then in the mass spectrometry part, there is also a huge difference between the different methods of how do you capture the spectra. It can be DDA, DIA, and so on. But I will not go into details on those things, because mainly I hope somebody else has explained that before. If not, there is plenty of documentation about that in the internet and in the course. What I am interested on is basically how quantitative analysis happen, and how this quantitative analysis can happen in a more distributed way. Then I will say that in order to quantify the final proteins, there is the proteomics data analysis can be divided in multiple steps. And the first one, for example, can be the protein database handling, when you create decoys or you generate some specific database, like custom databases if you're using RNA data and so on, to complement the analysis. There is another block which is related with the mass spec data processing, when you generate the pick list and so on. Then there is a huge block, and this is probably, you have seen some talks during the course about peptide and protein identification based on databases, including search engines and so on. And then there is a huge part in the quantitative part. Some studies, by the way, by only identifying the peptides and the proteins and the PTMs in the proteomics study, and the output of that is basically these peptides and this protein has been seen in this particular context. This is quite relevant, for example, in so-called proteomics experiments, when you try to identify variants at the peptide level and the protein level, and then identifications is enough, let's say, in some way to of the output of the pipeline. But if you're doing a quantitative experiment, for example, then you want to really quantify how much of this protein is present in one particular sample, one particular condition, and so on. And this process is basically start by trying to, if there is different type of quantitative experiments. I think someone also in the court has explained the type of label-it or label-free quantitative experiment. I will try to be focused on, in this particular talk, in label-free experiment. And in those experiments, for example, the quantitations happen in the MS1 spectra. Then there is a process where it's called feature map and feature detection, when you try to assign the intensity of one particular feature to a peptide. This process needs to go through all the MS1 spectra and all the MS2 spectra to try to identify for every single peptide what is the intensity of this particular peptide in the sample. And then the intensity is used as the proxy for the quantification. Let's say if you found that this peptide is more intense, let's say in the sample, then there is more of this particular peptide in this sample compared to another. Then what happened after that is then peptide needs to be mapped into a protein, and this process called protein inference. But actually, we try to assign all these peptides from where this belongs. There is a lot of algorithms and tools that does protein inference, and there is some challenge around protein inference because, for example, so you can detect peptides that actually are shared between different proteins. And then the algorithm, the major difference between the algorithms is how do you handle those proteins. If protein A contains the same peptide that protein B, which one is the one that you can quantify? And that's the protein inference algorithm try to handle that complexity. Then you translate that into protein abundances for each sample, on each sample, and then you perform a statistical analysis on the differential expression. Okay, then you say, okay, this group of proteins are expressed in this particular sample with this abundance, and then this set of and the same proteins are in another sample are expressed in a different rate. And then you try to compare samples and try to compare the expression of each protein on each sample to try to come up with some biological conclusion about how the expression of the particular protein is changing between conditions and so on. And there is sometimes it's missing a missing point, let's say in some way, where you try to visualize the quality control. It's how good your experiment has been. And this includes, for example, not only the final quantitative experiment, but also how the data was captured, if it was captured in a good way, if all the samples were captured in a similar way, if the number of spectra is similar for all the fractions, all the samples, and so on. And this is a particular this is how data analysis is performed. As you can see, there is multiple steps here. All of them can be even divided into more tiny steps that are complex in their own, and they consume time, also compute power in terms of CPU, memory, and so on and so forth. And today we will try to talk about tools that does all of these steps, and I will be particular talking about how you can distribute all this processing into a more distributed way. Then there is multiple tools that you can perform data analysis with, label-free data analysis. The more popular ones are LFQ, MPy, for example, and there is multiple tools that can perform data analysis. I will talk today, I will mention only in these slides the free tools, because there is other tools that I will mention in the next one that are basically proprietary tools that you really need to pay for that, they're not free. I would say the most widely used tool in proteomics is MAS1. You have to talk about that. But there is others like OpenMS, OpenSWAP, Skyline, Transproteomic Pipeline, Pattern Lab, Census, ProLine, MSBase, and so on. Most of them are, some of them can perform multiple data analysis. For example, OpenMS can perform SciLag, iTrap, TNT, and so on. And they are they have been developing multiple programming language. One of the major differences between those tools is that some of them are monolithic and some of them are non-monolithic. What is the monolithic tool? The monolithic tool is basically a piece of code, a software that performs all the tasks for you. And if we go back to our previous diagram where we show all the steps of the proteomics data analysis, then you will see that these are too many different steps in a monolithic tool. All of them are performed by the same tool, the same piece of software in the same machine at the same time. Okay. The benefit of that is that basically it's easier for the user basically to take all their experiment and analyze it in one big tool. Or there is another type of approach which is called a non-monolithic tool, when actually all these diagram and all of these steps can be divided into multiple pieces, and they can be distributed across different machines, different compute nodes, different compute power. That differentiation, or that splitting that the diagram can happen at the level of the tool, so sometimes also at the level of the data steps. Okay, then today, which ones are monolithic and which one are more tools that can perform the data analysis in one desktop machine? For example, MaxQuant, Pattern Lab, for example, are ProLine, for example, are tools that can perform the analysis in a monolithic way, where the other ones are more non-monolithic. As I said before, we did a benchmark 2 years ago when we actually tried to come up with which are the search engines, the peptide and protein identification tools that are more used by the community. And so far, the MaxQuant, for example, is the tool that and Mascot are the tools that are more used by the community, and within Mascot also Andromeda is the one that is more used by the community. This is in terms of publications. In terms of data analysis, I think the tool that is more prominent within the open source tool is Mascot, and then Skyline. But there is others like OpenMS that I will be talking today, and Philosopher and so on that also has a lot of attention by the community. And as I mentioned before, I will not talk about them, but there is also tools that are commercial tools that are quite popular, like for example Protein Discover that came with thermo instrument, and also Mascot, Progenesis, and Scaffold, and Spectronaut. Today I will be talking about OpenMS as an option or tool that can help the user to distribute the proteomics data analysis. OpenMS is a framework that provides more than 180 software tools, and mainly what it does is provide the infrastructure that you can run individual tools for processing your own task on your own data. OpenMS is mainly open source, is a computational framework, can be used in Windows, Linux, and also Mac. It's actually one of the most interesting points is quite based on PSI standards. It supports MCML, MCTAB, MCRNML. I think you have seen this this file format within the course, and also has been integrated with some workflow systems. A workflow system is basically a software tool that enables you to distribute tasks in more distributed environment. Let's say a computer cluster in HPC, a cloud environment, AWS. This kind of infrastructure that the tool doesn't run only in one big machine, but mainly in multiple specific machines. And these are the workflows engine allow you to take those specific tasks and tools and run them, get the result back, and go to the next task. And OpenMS, I think, is the most widely software in proteomics that has been integrated with this workflow system, especially with Galaxy M9. For example, the workflow that you see here is a 9 workflow. It's actually quite powerful for people that are used to use desktop applications. And as you can see, each task in a workflow is basically an individual task. For example, this is loading the MCML, this is loading the spectra, and this one, for example, is a metabolomics experiment, but those can be used for proteomics, is when you try to trace or detect features in the MCML files, and then finally also do retention time corrections. And these are 3 different steps that happen in 3 different that can happen in different times, but each of these steps is actually an individual task that only has an input and an output that goes and connects to the next step. Then a workflow would be like individual tasks that are all connected, and this gives an amazing flexibility for the user to connect and configure dynamic workflows, and you can replace tools and so on. I will put some examples afterwards. Enabling the user to perform really nice experiment when in a monolithic tool you basically put your data and get some results after that. But in the monolithic tools, only a software is used for protein identification, a software is used for feature detection, and only a software is used for protein inference and so on. In these particular workflows, you can replace, for example, tools to perform the protein inference by another tool, which gives you more flexibility and more power over your data. For example, this is a simple peptide and protein identification. Then you have the input data, and this one, for example, are the MC file, the data that contain the spectra, and this is the FASTA file. And then all the data goes into, this is an OMSA adapter. This is basically the search engine of OMSA that has an OpenMS, it's an OpenMS tool, and the user can use that. But it's easily replaceable by other search engine. You can try and say, okay, I don't want to use OMSA. It doesn't give me results. It's a quite old search engine. I want to replace that by a new search engine, Comet, which for my data give me more results. Then you can easily replace this box by another tool. This is also quite powerful because when new tools are created, then OpenMS framework easily create a tool for that, integrate that into the framework, and then in 9, for example, you can replace that by a new tool. Where if it's in a monolithic tool, you really need to wait if the tool is supported, and most of the monolithic tools only support one search engine, the one that belongs to the monolithic tool. For example, MASQ is Andromeda, do not allow to bring new tools into the workflow. And then after that, as I said, there is multiple steps that always happen in a protein identification. You need to, when you identify the peptide, you need to map those peptides into protein. That is what is called peptide indexer. Then you need to do the protein FDR calculation, filter those the peptides that you're not interested in, depending on the FDR steps and so on. I need to say that, for example, if you're using a monolithic tool like, for example, Peptide Checker or MaskOne, in this step, you really need to, these steps are all together in one place. Then you load your data, your FASTA file, and you say, I want my data 1% FDR. That's it. In this type of workflow, you really need to build your steps and provide the parameter for each independent step, which at the very end you will have the results, the final result. You will be performing the same task, but you need to really build your workflow yourself. The best tool to visualize the data in output data from OpenMS is TopView so far. There is other tools like R packages and so on that are quite well integrated with OpenMS, like MStats. The workflow, as I said, is a plugin system, and I will, going back to the previous pipeline that we have only a pipeline that has an OMS adapter, for example, but now in this particular example, we just added a new search engine. We just say, okay, I want to also put here extended search engine, okay? Because in my analysis, I have more computer power, and also I have, I want to increase the probability to identify my peptides by using another search engine, and compute and combine the data coming from two different search engines to basically have more probability to identify my peptides and identify more proteins and finally quantify more proteins. Then if you identify more, if you can easily add a new search engine, in this case would be extended, but of course there is some extra nodes that needs to be added in order to combine two search engines, and these are the two the new nodes that you need to add are, for example, file merge. You need to first compute the posterior error probability, because the search engine came with their own search engine scores. This needs to be translated into a common score. Then you need to merge the files coming from the two search engines and then produce a consensus ID. After the consensus ID, all the steps are the same. Then what I am saying here in summary is that I will explain a little bit more about small nodes and so on, but the message to take from here is that you can really customize complex pipelines to analyze your data, and this become more interesting where more complex use case wants to be done, like proteomics experiments, or when you want to really go deep, identify more peptides with using multiple search engines and so on and so forth. For example, in 2017, OpenMS teams, and I was working with them, and we did a really big benchmark of multiple search engine, combined all of them with multiple protein inference to see which one give us more results. I mean, it was published in Journal of Proteomics 2017, as I said, and then we compare the combination of different search engines. How do you improve the number of peptides and proteins identification? For example, if you use only one search engine, for example, extended, you can get 1,200 for that particular data set, 1,200 proteins. But if you use MSGF plus, you get around 1,600, and then when you combine them, what the results are, some of them are half overlap, especially at the protein level. I will not go into detail. At the protein level, you don't see that much of results of improve, but at the peptide level, you get more coverage for each protein because you can have more peptides identified. Then some of the identification nodes, as I said, are ID posterior error probability that convert basically search engine scores into a common score, fault discovery rate to compute the fault discovery rate, consensus ID to combine PSMs. And there is other, what I call complementary nodes, and these nodes are more for data handling, like for example when you want to merge search engines and so on, or when you want to for example, peptide indexer take the output of the search engine peptide and try to map them back to the protein sequence to know from where proteins come, these peptides come from. And for example, ID filter, which allow you to perform filtering operation in the peptides identification output, like for example, filter by FDR or even for peptide length or other features. There is other more complex workflow, like a label-free workflow, and they becomes bigger and bigger. I will not go into details on that, but there is multiple publications around different workflows of label-free based on OpenMS, and all of them has been basically the difference between them are basically how do you combine or add your own algorithms into them. How this is how basically it happens is there is not a big unless you use KNIME as the desktop tool to build your workflow, you can run this in the command line. And I think the best way to use OpenMS tools is to combine it into pipelines. And a pipeline and workflow is basically a piece of software that allows you in the command line to run in a cluster, in a cloud environment, your data analysis rather than in the desktop application. As I said, OpenMS is a modular workflow system and supports SciLab, ArcTrack, TNT, label-free, and so on, and has a strong collaboration with search engine, HUPO, PSI, Percolator, and so on. These are the links to get information around OpenMS. But I will talk in the last part of the talk, even if OpenMS allow you to build your workflow and run your workflow in a distributed environment, by itself, OpenMS is not a workflow system. It's basically a framework of different tools that can be combined together to build a workflow, and you need a workflow system to basically build, to run your workflow. KNIME is one of them, the one that I showed before in the slides. KNIME is basically mainly a desktop workflow system. What a workflow system basically allows you to take that task that you have defined, let's say identification of this MCML, of this mass spectra with this software and this database, run that particular tool in one machine and get some results back. This process of sending to a machine, get the results there and retrieve from there into one place. This is the task, and then send the data output of this particular task or node into another step. This is what the workflows allow you. Last year, myself with Pablo Moreno also, we published a really nice review and opinion manuscript around how workflows can be used, scalable workflows can be used in proteomics, and what are the challenges, still the challenge in proteomics and metabolomics data analysis. What you really want is basically that all the analysis steps of proteomics workflow, let's say database searching, FDR filtering, protein invariance, RT alignment, feature detections, and so on, can be combined, can be split into a small task. This small task goes into a tool, and this tool should be going into a container, for example, the Docker container. And a Docker container is basically a self-isolated environment that can be run and deployed into a machine. And finally, this particular tool can be run in different machines and then get back the results and combine the results. A A workflow system basically allows you to take all these tasks, create tools for them, and deploy those tasks, combine those tasks into a workflow or a pipeline. Sometimes some people call it pipeline. And the workflow system, what allow you is to deploy and run those tasks into different type of environment, including a local machine. Okay, then if you have a workflow, it doesn't stop you to really in your machine, in your Mac or in a mainframe, deploy and run your workflow. But mainly is the desirable architecture is basically the cloud architecture. For example, cloud or HPC architecture, where distributed architectures in general, when you can actually send each specific task into one node and get back the result. What is the best? What is the benefit of that? There is multiple benefits. For example, if you are running your whole analysis in just one machine, well, you expect that it will take more time. That's the first thing, because if you split in multiple tasks, those tasks can be sent to multiple machines, okay? Then you have more compute to basically analyze more data and finish that analysis in less time. That's the first benefit, but there is others. For example, imagine that you, for example, are analyzing, you're performing database search into 20 different files, and those one file is failing because of needs more memory or something like that. Then instead of waiting 1 week for your server to arrive to that file, if you're in a single machine analysis, to arrive to that file, to perform the data search, and then find out that you really need more memory, that's when you're analyzing just one machine. When you're analyzing multiple machines, then all of them can be distributed, and in a matter of minutes, you will receive a message, okay, a new machine needs to be allocated for this particular file only. And then you can reallocate that particular data analysis into a bigger machine and get back the results and move to a smaller machine. Then there is also a resource, in terms of resource, it's actually better because you can really allocate a small task to small machines to finish quick, and bigger tasks to big machines to also finish quick. And that's one of the also benefit of distributed cloud. There is more and more benefits. Today, myself and OpenMS team also, we have been working in collaboration with Nextflow. Nextflow is a workflow engine. It's a framework that is used heavily by bioinformaticians in genomics, transcriptomics, and so on, that allow you to incorporate your bash scripts, your pipelines into a more reproducible environment, scalable, and also with a lot of checkpoints in the middle. It's reproducible because it uses Docker and Singularity container. It's portable because you can deploy it in Google, Kubernetes, AWS. And you also use parallelism. You can basically distribute tasks across nodes and then get back the results to other tasks, and has a continuous checkpoint, stream-oriented, and so on. Then we recently developed actually a really nice workflow called Proteomics LFQ, based in Nextflow with OpenMS tools, that takes the input of the workflow that I was explaining before for protein quantification, and actually split that into a small task. Then you have the database here, and then you can have also the MCML or raw data here as an input. And then you have the SDRF. I don't know if someone talked about this in any of the presentation, but the SDRF is a new development that we have done, where we actually provide a way of describing the sample, but also some of the parameters of the experiment, like, for example, PTMs, precursor and fragment error tolerance, and so on, enzymes, and so on. Then the data is actually, there is one step here for the FASTA file, like it's called decode generation, is optional. And then there is a raw conversion you take the raw data from the instrument and then convert into MCML. Here you can use two search engines. Now are supported only two search engines, but there is another one coming called MSFragger that we can actually plug a different way of performing data analysis using OpenSearch. Then all of the data is actually mixed, and we use Percolator to boost the number of peptides. Percolator is a machine learning tool that takes the peptides identification coming from the search engine and try to increase the number of ID by trying to differentiate between true positives and false positives using other properties like retention time, length of the peptides, and so on. If you want to really improve the number of IDs in your tools, you use some sort of approach like Percolator. There is others. Then the FDR is then the data coming from MSDF and Comet are combined, merged. As I said, these are all tools coming from OpenMS in a consensus, and then combined FDR is performed, and then the peptides are filtered by the FDR filter can be 1% FDR or and so on. It depends on the analysis that you do. And we have a really nice tool on top of that. If you're doing phosphorylation, phosphoproteomics data analysis, we use Lucifer to actually try to give a probability that one specific site in a peptide, a phosphosite in a peptide is true or false, and then give you a probability that this the correct site for each phosphorylation. Then on top of that, there is a big step. For example, all of these steps can be run, let's say, in some way in parallel. I will explain what means that. But there is a final step that performs the protein inference, because the protein inference, it's a step that needs all the data coming from all the samples and actually perform the analysis in all the data together. Then that will be, it can be seen as the big and heavy step of the whole process. What the workflow does actually is basically taking all of the MCML files, is a label-free experiment, and run them independent in these independent tools. Then here, for example, this is how you can run after you run the command line. This is run in a cloud environment. What actually the Nextflow is doing, Nextflow is basically taking, for example, in this case, we have 5 particular raw files. It's not this SDRF, it's another one. It has 5 particular raw files and perform the conversion, this conversion step. Each of these conversions has happened in a different machine in parallel. That means that if you have a big frame, you need to convert all of them one after another. In this particular workflow, you're sending the 3 files to the 3 different machines, let's say, and then convert it at the same time, which you can see as you can try, you reduce the time of performing each task, but the number of compute that you can allocate. And this becomes really relevant when you add more and more and more samples. Of course, for 5 samples probably doesn't make a huge difference, but if you have thousands of samples, thousands of raw files, they can be all of them distributed and get back. Then you see here in the search that you have performed MSDF plus, which is one search engine. You still haven't finished any of them, and this is a really nice point. And Comet actually, which is the other search engine, has finished the 5 tasks because it's faster than MSDF plus. Then all the steps that are related with Comet has finished. Okay, the next step for Comet, which are, for example, Percolator on top of Comet, the distribute based posterior error probability calculation, all of them has finished. But the next step that combine the result with MSDF plus, this one here, hasn't happened yet, because the PyLam is actually waiting to happen all the MSDF plus to start again all the process for that needs the output from Comet. This is how the workflow engine works. The workflow basically sends tasks and only wait for and only when one task and the other basically need to be converged into one particular task. Then there is a huge advantage of using Nextflow. We'll not go into detail here, but you can actually trace the use of CPU by each individual task. You can trace the RAM, the resource usage, the CPU by each individual task. And for example, you can see here from my pilot that MSDF Plus, for example, is taking more physical memory here. You see here where actually the other search engine is not taking almost any memory, but in terms of CPU, for example, here Comet is using more the CPU than MSDF Plus. Proteomics LFQ also has a really nice tool called multi-QC that generates reports around the peptides and proteins identified. For example, how many peptides have been identified with Comet, with MSDF Plus, and the final number of peptides when you combine them. As I said, we also have another tool that is using MStats to actually combine the result from multiple from the proteomics LFQ into one simple R-Shiny application that you can see, for example, the volcano plots, which are the processes that are changing the expression and how. Thanks a lot for the opportunity to present how you can actually perform distributed data analysis into in the cloud and also in HPC environments. And I hope to get your questions in the course.